Olá, bom dia a todos, bem-vindos ao seminário ao webinar, eu deveria dizer, do Instituto Serrapilheira, que se organiza conjuntamente com o ICTP-SAIFR, que é na Unesp, em São Paulo. Hoje estamos muito felizes de apresentar um webinar da Akiko Iwasaki. Eu vou apresentar ela daqui a pouco em inglês, porque eu acho que é melhor dela entender. Eu quero só lembrar que esse webinar é também uma ocasião de lançar um curso de Biologia e de Ecologia Quantitativa que o Instituto Serrapilheira, juntamente com o ICTP, vão lançar. Esse curso vai acontecer em julho desse ano e as inscrições vão ser abertas na próxima terça-feira, dia 9 de março. Então é importante, quem quer se inscrever, por favor, esteja prontos no dia 9, porque nós vamos dar preferências aos primeiros 500 inscritos. Então é importante ficar tudo preparado, vocês podem ir no site, está tudo ali, vocês podem ver o endereço do site. Se vocês têm pergunta, tem um e-mail também, vocês podem fazer perguntas. Por favor, eu acho que é uma grande oportunidade de fazer um curso realmente bem legal que vai acontecer em julho. Nós também estamos à procura de tutores, são pessoas que já têm um doutorado e que vão acompanhar os alunos durante o curso. Então, se vocês têm interesse para ser tutores, por favor, também está tudo no site, vocês podem achar as informações para inscrever. Agora eu vou falar em inglês para que a Akiko consiga entender. Akiko, I'm switching now in English. Very, very happy to have you here today. I was just reminding that this webinar is also an occasion for us to launch our new program, a new training program on quantitative biology and ecology, and it's a great way to launch, right? So we had a seminar from Simon Leving on Tuesday, and today we have you. So Akiko Iwasaki is a professor of immunology at Yale University, and she's a very famous professor of immunology because she's been extremely active recently in the COVID crisis, and I guess she's going to say more about this during her seminar. You should know that she's an HHMI investigator. She's been extremely productive. She's been publishing. I'm not going to list all of the papers that she's published because that will take way too long. But she's an extremely productive scientist, and she's a modern scientist in a way that she's been extremely implicated in defending the role of women in science, and those are values that we are really dear to the Serrapilheira Institute. She's also been very much a defender of the well-being of students, PhD students and postdocs in the lab. We know that sometimes it could be tough, and so PIs have to do their share. And she's extremely active on Twitter, and I don't know if you follow her, but she's actually recently tweeted in Portuguese, so I want to really thank her for that. She's been really, and so we're very happy to have Akiko with us today. She's a reference in the field of immunology. She's been working specifically on the role of mucosas in the immune response. She's been working on various viruses. I don't have time to say everything that she's done today, but of course she's been extremely active on the COVID-19 crisis in the last year. She's part of the advisory board of the training program that we're launching today. So we are very happy to have her today. And I'm not going to take more time. I'm going to hand you the mic, Akiko, and I want to really thank you very much in the name of the Serrapilheira Institute and of ICTP as well. Thank you very much for being here. I just want to tell the people who are listening that you can ask questions in the chat. please ask your question. At the end of Akiko's seminar, we'll have a little bit of time, and I will ask her the questions that you send us. So please feel free to ask questions. Akiko, thank you very much. I hand you the mic. We are looking forward to your seminar.
Thank you so much, Hugo. It's my pleasure to be here. I'm so excited for the launch of the new training program in quantitative biology and ecology, and it's my honor to be speaking on the launch. So I will share my screen. Okay. Can you see? Okay, great. So Hugo, you asked me to talk about the important role of basic science in attacking the real world problem, which is the COVID-19 pandemic. So I put together slides to be able to highlight that. And it's really important to keep in mind that all of the amazing feat that science was able to accomplish in the last year or so really is based on basic science that's been, investigated for decades prior. So the outline of my talk will be to give a brief overview of the virus itself, the seasonality, and the organs involved in this disease. And I will give you one example of our own work where we characterize the immune response during severe and moderate COVID disease. And I will also talk about the vaccines, really the highlight of our success in this pandemic. How do they work? Are they effective? What do we know? What do we not know yet? And then I will conclude with a couple of slides. Okay. So SARS-CoV-2 is the causative agent of COVID-19, and it's a member of the coronavirus family. And throughout the talk, I'd like to highlight women in science that have contributed greatly to our understanding of virology and immunology and have yet have unfortunately been kind of ignored and under-recognized. So this is one of those. First example is Dr. June Almeida, who is the first person to have really identified and recognized the crown structure that gives the name corona, the crown virus. And she was an expert in electron microscopy, and she was the one who identified these crown-like structures. And later on, we now understand that those crowns are made of these spike proteins that protrude out of the viral envelope and are used by the virus to attach to the host cell to enter and replicate. And in addition to the SARS-CoV-2, there are two other zoonotic coronaviruses, both of which cause severe to lethal diseases in humans, including the original SARS virus, as well as MERS-CoV. And there are, in addition, 4 other more regular seasonal coronaviruses that cause common colds. Up to 30% are thought to be caused by the collection of these kinds of coronaviruses. So humans aren't completely new to the coronaviruses, and yet this particular virus emerged recently in 2019 that we really didn't have previous immune response to protect us. So what do we know more than a year into this pandemic? From prior research, we knew that people who have recovered from common cold coronaviruses can get reinfected again with the same virus within about a year. And we have seen multiple evidence of reinfection with SARS-CoV-2. And so the natural immunity to this virus isn't perfect. And so the vaccines, in contrast, appear to be providing very protective immunity against infection. And that's what we really need to achieve herd immunity, which protects the entire population against this virus. And we must kind of be vigilant about these variants that are on the rise that appear to have enhanced transmission and ability to evade antibody responses that were developed from the original virus. And thus, affecting somewhat the efficacy of the vaccines. And I'll get to that later on. So as with other coronaviruses, there is a seasonality to all respiratory viral infections that commonly we study, such as the influenza virus and rhinovirus and others. And for several years, we've been interested in why the season affects viral infection and transmission. And so we have been studying a variety of these seasonal factors that are indicated here, such as the temperature, the humidity, and the vitamin D status. And it's known that these factors directly affect the virus itself because it alters the viability of the virus, as well as the droplet dynamics, the size and the matrix, and how efficiently they transmit from one person to another. We focused more on the host defense side of the equation and found that a lower temperature that's found during the winter time in the nose, for example, can impair innate resistance against rhinovirus. And we've recently shown that low humidity that's found in the indoors during the winter months also impair the ability of the animals to combat influenza virus. So there is, in addition to these sort of physical impact of these factors on the virus, it also affects the host defense. So this is something that we're continuing to investigate for the SARS-CoV-2. So COVID-19 is transmitted through the respiratory route, but is now understood to be a kind of an extensive involvement of multiple organs throughout the body. So its impact on the lung has been, the primary focus of many studies. And we know that the virus is inhaled and it can infect the epithelial cells of the lung, and it can from there cause damage as shown here. However, it also impacts multiple other organs, for instance, the blood vessels in the heart, as well as the brain. A lot of people having neurologic symptoms after infection. The eye, the nose, the liver, the kidney, intestine are also impacted by the virus. So it is not a kind of a simple respiratory infection, but we really need to think about the systemic impact of this virus infection. So it's really difficult to give you an overview of the immune system, but I tried to do this in one slide, just because I think it's important for us to continue talking about, what I'm about to share with you. So the immune system is divided roughly into two arms. One is the innate immunity, and the other is the adaptive immunity. Innate immunity is something that is intrinsic to every cell type. So when a virus invades a particular cell, this virus is recognized by sensors that are dedicated to detecting features of virus, and that triggers interferon responses. These interferons are molecules that are secreted out of the cell to bind to its receptors. Essentially, every cell type can detect this interferon, and signaling the neighboring cells to put up barriers so they are no longer infectable. And also signaling other cells that are infected by the virus to die, because we don't want these viral factories to go on. And also recruits other white blood cells to the site of infection to try to help clear this virus infection. This occurs within minutes to hours of infection, and it's really key in controlling the viral load over time. If this interferon response isn't triggered properly, then a host cannot control the virus very well. On the other side, within about a week or two, we start to see this adaptive immunity. And the word adaptive really means that it is really specific to the particular pathogen. And there are two different kinds of white blood cells known as T lymphocytes and B lymphocytes that contribute to the clearance of the virus during the adaptive immunity. And these lymphocytes are educated so that they detect specific element of the virus, and kill the infected cells in the case of killer T cells. And B cells can secrete antibodies that are required to block the transmission of virus by binding to the surface of the cell. So this takes about a week or two because each lymphocyte that's specific to these particular viral antigens are extremely rare. So you have to first find them, and then you have to expand them. Then you have to educate them, and they have to circulate around the body to find the infected site. But once that happens, they're really great at controlling viral replication. And the adaptive immunity is really the reason for any vaccines working. So we really want to educate the immune system, the adaptive immune system, to trigger these powerful resistance mechanisms. But it turns out that the innate immune response is key to trigger this adaptive immunity, because the antigen presenting cells that allows for the education of adaptive immunity has to trigger its own innate immune signals for this to happen. So I'll get to that when I talk about vaccines. So I told you I'm going to share one story out of the lab, which was really investigating how immune responses are mounted against the SARS-CoV-2 virus when people are infected and have severe or moderate disease. And so it was led by Carolina Lucas, who is a postdoc, came from Brazil, and she is also working with 3 other key investigators who co-led the study that I'm about to tell you. So in order to study immune response, we can't just have one time point from one patient, because we don't know what happens in a sequential manner within that individual. So what we did is we recruited patients into the hospital who are in the hospital who have COVID into the study, and then longitudinally collected their sample every 3 to 4 days until they were discharged from the hospital. And we basically divided the patients into moderate versus severe, depending on the amount of oxygen that they required during the hospital stay. The least clinical score, number 1, did not require any oxygen, whereas number 5, for example, required mechanical ventilator, and then number 6 is death. And we also had a recruited uninfected control, which are the healthcare workers that were collected at the same time. So what we did with these is we collected their blood, the nasal and oral saliva samples, and then over time see what happens to the virus and what happens to the immune response. So the first thing we notice is that, when you collect blood from a healthy person, the composition of that blood, you can see there are T cells and B cells and NK cells. These are all white blood cells that are important in the immune system. And the percentage of these blood cells are indicated here. This is a typical percentage of what you see in the peripheral blood mononuclear cells. With the COVID patients, though, what we noticed was that there was this quite significant reduction in the percentage of the T cells, and also increase in these cells known as monocytes that usually patrol different parts of the body for infection. So we're seeing elevated level of that. And we also started seeing things that we normally don't see in the peripheral blood, mononuclear cells, which are these so-called granulocytes, because they have granules inside them, known as neutrophils and eosinophils. And they were actually coming into this particular fraction in severe to moderate COVID patients. And when we looked over time, what happens to the viral load in these patients, what we saw was that the patients with the severe disease, indicated in pink here, were unable to control the viral load in the nasopharynx over time, whereas those who had moderate disease were ultimately able to control the viral load. So this indicated to us that maybe the lack of clearance of the virus is really what's leading to the severe disease. And this hypothesis is supported by the fact that there are many cytokines that are correlating with viral load. So in this case, we're plotting the viral load on the x-axis, and on the y-axis, we're plotting different kinds of cytokines we see in their blood. And you see that the interferon alpha, which, as I mentioned, interferons are the antiviral factors that are key for innate resistance, is actually elevated as a result of having higher viral load. And this was strange because we expected that the higher the level of interferon, the less virus you would have. But it was the opposite in this case. So somehow this virus is able to evade the effect of interferon, but still able to trigger the secretion of interferon from the infected cells over time. The same kind of thing was seen for another kinds of interferon gamma, another inflammatory cytokine, TNF and TRAIL. So it really looks like that the higher viral load is driving all these inflammatory and antiviral cytokines. And when we looked in the patients that unfortunately died of this infection, which are the pink dots here, and compare the level of interferon alpha over the first 12 days of symptom onset, so fairly early during infection, what we saw was that they were elevated in the people who died of this infection compared to those who were discharged alive. These are the patients who went home after recovery. And similarly, we saw elevated levels of IL-1 receptor antagonist, which is sort of a mark that there was an inflammation that was going on in these patients during the first 12 days. And we saw similar kinds of trends for other types of cytokines, indicating that, this elevated interferon is not helping these patients, but potentially harming these patients. And they're enriched in the people who died of this infection. And when we looked across all kinds of measurement that we did, what we found was this very strange feature associated with COVID, in that not only do we see these antiviral type 1 immune responses being elevated and maintained high in the severe patients, again, all the pink ones are the severe patients, we also started to see type 2 and type 3 immune responses that are usually reserved for parasite infection and fungal infection. So this is strange because when I usually teach my students immune response in my course, this is the picture I give them, which is that for each kind of infection, such as parasite, viral, bacterial, fungal, we generate the right kind of immune response against these types of infections. And they're driven by the interaction between the antigen-presenting cell and the T cells. And the fate of the naive T cells to become these kinds of effector lymphocytes really depend on the instruction by the dendritic cells or the antigen-presenting cells, what kinds of cytokines they secrete to educate the T cells. But as I just showed you in the severe COVID, all of these effectors are induced at the same time in these patients. And how would an anti-parasite immune response help a viral infection? It's very unclear. And so we think it's a maladaptive response to the virus, and it's not really helping the patients recover from the infection. And when we looked at a variety of factors that most associate with mortality, what we found was, again, the interferon alpha being the number 2 biomarker for mortality, following IL-18, which is, again, a very inflammatory cytokine that is being seen in the severe COVID patients that could induce damage within the lung. So to conclude this one story from my laboratory, what we saw was there is a persistent viral load that's detected in severe patients, and that viral load appears to be driving interferon responses. And there is an increase in interferon and inflammatory cytokines during the 12 days of symptom onset in people who died of this infection. There are signatures of proteins and genes that were able to distinguish people who recover versus who develop worse disease. And in patients who developed worse disease, they had essentially all types of immune responses engaged, again, perhaps being a maladaptive response to a virus. And so this was just to give you a flavor of what we can do in a very short time to understand how patients are responding to the COVID. And so now we know that, a large variety of disease outcomes are possible from this one infectious agent, SARS-CoV-2. And now I just told you about moderate versus severe cases. There are people who also recover without symptom, asymptomatic, or very mild symptoms, and that tend to be younger population. There are also acute disease and long haulers. Long haulers are people who suffer from long-term consequences of COVID-19. And people have reported, over a year of symptoms, including neurologic and respiratory, GI tract, multiple organ systems that I showed you earlier. And so the host status, age, sex, comorbidity, obesity, multiple factors contribute to the outcome differences. And we also saw that sex differences in immunity might also account for some of the differences in the outcome that we're seeing. The male sex is a risk factor for severe COVID and mortality from COVID. So there's a lot to unpack still. We are particularly interested in what we can do to help the long haulers. Now that we have a great vaccine to prevent the acute disease, can we now focus on how do we deal with these? How do we help the long haulers? So I wanted to emphasize that the research that I described to you that we were able to do very quickly is really based on the foundation paved by the basic science discoveries that have led to the science today. So every technique used in our study was developed previously by numerous scientists trying to probe nature. This includes mathematics, techniques that were developed for different reasons that really became handy for biologists to use. And that's why this quantitative biology is an important aspect of even clinical translational immunology. Machine learning, we use that to segregate patients that recover versus those who went on to develop worse disease. We use flow cytometry, which is a technique that we immunologists often use to label cells and understand, what compositions of cells look like. We did PCR to be able to understand how many viral particles there were in the nasopharynx. ELISA to measure antibodies, and fractionation to be able to, study these PBMCs. So all of these techniques, we didn't invent this in our lab. It really existed because of huge investment by multiple people before us that have led us to where we are today. And so the basic science methods and sort of concepts are really important in revealing clinically relevant insights that could lead to direct paths to diagnosis, treatment, and prevention. And so I'm going to tell you an example of that with the vaccines. So moving on to sort of immune protection against COVID-19, I just told you what went wrong with the immune system during a disease, but we can also use the immune system to protect against disease. And so I want to spend the rest of my time talking about that. So this is a figure taken from a paper published by another group, Dunberg's group, where they measured what correlates with protection against disease or infection using rhesus macaque as a model of COVID-19. And so what they found was, so they infected a whole bunch of rhesus macaque monkeys, let them recover, and they challenged these, recovered monkeys with another dose of SARS-CoV-2. And they found that some of these monkeys were protected and others were not. And those that were protected had higher levels of these neutralizing antibodies, which means that neutralizing antibodies are a correlate of protection based on this primate study. And also, they were able to take the same kind of convalescent monkeys, and they depleted CD8 T cells, which are the killer cells that I told you about, from these monkeys. And then they challenged these monkeys with SARS-CoV-2 intranasally. And what they found was that, these lines indicate the amount of virus that they were able to measure from monkeys over time after challenge. What they found was that when they depleted the CD8 T cells, there was an elevated level of the virus that you can find in the nasal cavity, meaning that the CD8 T cells that were primed during the first infection was important in controlling the viral load within the nasal cavity. However, in the lung, there was very little difference whether the monkey had the CD8 T cells or didn't have the CD8 T cells, indicating that perhaps other features like antibodies are able to clear and protect the lower respiratory tract. So this really indicates the intricacy of different arms of the immune system that protects the host, circulating antibodies, as well as local T cells. And for all vaccines and protective measures, we should really be thinking about immune correlate of protection. And so speaking of vaccines, vaccines are already shown to induce robust antibody and T cell immune responses. This is a figure I took from Dr. Florian Kramer's review on the vaccines against COVID-19. And here are some of the vaccines at that time already, which had the phase 1 and phase 2 trial data. And this is really looking at the neutralizing antibody titer after the boost, as well as T cell responses if they were measured. And so you can see that many of these vaccines were able to induce medium to high level of neutralizing titers, and able to induce some T cell immune responses when they were measured, indicating very promising approaches to protective immunity. And so there are so many vaccines that are either already in emergency authorization use or in phase 3 trial, phase 2, phase 1, variety of stages. And they come in different flavors. So I just wanted to tell you that the vaccines are not all equal. Some of the vaccines are using the nucleic acid information of the virus, such as the mRNA vaccine, to trigger immune response against whatever the antigen that's encoded by the nucleic acids. And so the mRNA vaccines advantages are that it's very easy and quick to design. And if there are variants, you can easily encode the antigen from the variant to make a suitable vaccine that would prevent the infection and disease from such variants. Live attenuated vaccines and inactivated vaccines, these were sort of workhorse of vaccinology. I mean, this has been used for a long time to protect humans against a variety of diseases. Live attenuated is not currently used in the case of COVID-19, but inactivated vaccines have been developed by multiple different groups and companies, and some of which are already approved for use in humans. There are subunit vaccines. These are sort of protein subunit. So for instance, the spike recombinant spike proteins are used by some companies to develop vaccines. And this is, again, a traditionally used method for stimulating the immune response with a vaccine. And then we also seeing multiple viral vectored vaccine. These are sort of, using a viral coat from another virus, like adenovirus, and then encoding within the genome of the adenovirus, the spike protein from the coronavirus to make a kind of a recombinant chimeric vector vaccine. And this allows the cells to become infected using the coat of the adenovirus and then inject the material into the nucleus of that cell. And that cell becomes a factory for the antigen, which is in this case spike protein. So there are advantages and disadvantages of all of these approaches. But the good news is that all of these vaccine approaches appear to be working very well for SARS-CoV-2. So I want to highlight this new kid on the block, the mRNA vaccines, today, because it has really had one of the most spectacular success and the quickest to reach the human use, at least in the US. And so the mRNA vaccines, again, really is based on decades of work by multiple investigators. But I wanted to highlight Dr. Kariko, who has been really struggling to sort of stay within the sciences because of variety of barriers that she faced during her career. But here is just an excerpt from the Wikipedia that describes that, she was born, she's a Hungarian researcher who came to the US to pursue her dream of using RNA as a mechanism to stimulate the immune system as a vaccine strategy. And this path has not been easy for her. She was denied being a professor, tenured professor at the University of Pennsylvania, because of sort of lack of application and success during her tenure when she was there. But she found a collaborator, Dr. Drew Weissman. Together, they developed this RNA vaccine strategy. So if you just take the mRNA and inject it into a cell in vivo, that would cause a lot of inflammation, as I will get to. But Dr. Kariko really figured out how to evade that inflammatory response so she can use this mRNA as a vaccine platform. And the rest is history, essentially. Like, it's an incredible vaccine platform that I think is going to change the nature of vaccinology, forever. But anyway, I just wanted to highlight her because not much credit was given to her throughout her career. And only until very recently, she was recognized as a leader who really led the field forward so that we have a COVID-19 vaccine today. And she is currently with the BioNTech team. So, I'm just going to give you some figures from this really nice review on mRNA vaccines that was written by these authors. So as I mentioned, the mRNA, if you just injected it into an animal, would cause severe inflammation, and it's just not tolerated. So over the many years of multiple iterations of effort, Dr. Kariko and others have really figured out how to make this the mRNA vaccine competent. And so here are some of the list of things that she tried and made mRNA less immunogenic. And also better at being translated and generating the antigen of choice. And so I just show you a picture of that. So an unmodified, unpurified mRNA, if you was just injected into the host, would trigger all kinds of viral sensors. So in the beginning, I told you that every cell is equipped with sensors that can detect viruses. And indeed, that's what the mRNA looks like. mRNA that's taken into these endosomes where it shouldn't be there. mRNA is usually in the cytosol. So when it's in the endosome, it triggers this toll-like receptors that are usually there to detect viruses. And this induces all kinds of interferons and cytokines and all the things that a host is supposed to do when they encounter a virus. But it's very toxic because you're basically stimulating this throughout the body, and it's not tolerated. So it wasn't a very good vaccine platform when she first started doing this. But then after multiple modification and also carriers strategy, so every component of this mRNA vaccine is important. Like, how do you package the mRNA? How do you deliver it to a cell? How do you avoid innate sensing? How do you trigger the right kind of cells to be taking up this vaccine? In any case, by introducing the pseudouridine instead of the uridine, which is naturally found in the mRNA, she was able to kind of coat the mRNA in such a way that the toll-like receptors are no longer able to see the mRNA very well. So this allowed the mRNA to go into the cell from the endosome into the cytosol. And this step from the endosome to the cytosol was enabled by the lipid nanoparticle technology that was developed further. So every step of the mRNA vaccine had been optimized for proper process production of the antigen as well as delivery mechanisms. So anyway, she was able to convert this inflammatory thing into a very tolerable and effective, effectively translated mRNA. And as I mentioned, you can't just put the naked mRNA, even if it's pseudouridine and optimized, because it doesn't get taken up by the cell very well. So there are many, many formulations that were tried, indicated here, nanoemulsion, dendrimers, and different kinds of liposomes, and so on and so forth. But the lipid nanoparticle that are shown here are the ones that are now used for human vaccines. And they seem to be extremely good at delivering the mRNA into the cell for immune activation. So how does mRNA vaccine work? Well, you take this pseudouridine containing mRNA with the optimized lipid nanoparticle, you inject it into a person, into the arm, and that particle is now injected into the tissue and are taken up by these dendritic cells or antigen presenting cells that I showed you in the very early slide are the ones that educate the lymphocytes to become activated and are specific for the antigen, in this case, spike protein. And so the dendritic cells, antigen presenting cell, take up the nanoparticle, express the spike protein, they migrate to the lymph node, they stimulate the adaptive immune response, and that's how this antibody responses and T cell responses are generated after vaccination. There are many clinical trials before COVID that have tested this mRNA platform for a variety of diseases like HIV, rabies virus, Zika virus, influenza, and so on. But until now, there had been no approved mRNA vaccines. But obviously, with the COVID on the horizon, the team of BioNTech as well as Mrna really accelerated their development and are now obviously approved for use in humans. And this is really a remarkable slide that shows the efficacy of the Pfizer-BioNTech two-shot COVID vaccine. Phase 3 trial results showing somewhere like a 95% efficacy from preventing symptomatic infection 7 days after second dose. And, remarkably, it's also able to completely prevent death and mostly severe infection. I think this is also probably 100% protection. And you look at another mRNA vaccine, such as Mrna, and again, the efficacy is almost comparable, 94% from symptomatic infection. And then if you look at the severe infection and death, there's 100% protection against those. And then if you look at the vectored vaccines, such as the AstraZeneca 2-shot vaccine, you see that depending on the kinds of doses that were given, you can see that the efficacy varies. But again, hospitalization from COVID was reduced to 0. This is another vectored vaccine, adenovirus Ad26, single-shot Johnson vaccine. And again, if you look at the severe disease and COVID-related death after 28 days or 49 days of single shot, it's 100% protection. And again, the moderate and severe diseases, the efficacy differ between different countries, likely because of the rise of the variants being able to evade some of these protective mechanisms. But from all the variants, there are no COVID-related death yet. This is a recombinant spike protein vaccine by Novavax. And again, the phase 3 result looks great, 89% protection from symptomatic infection. And, if you look at the South Africa sites, the efficacy reduces perhaps because of the variants. But again, against all variants, there are no death or severe diseases. Finally, this is the Sputnik 5 Gamaleya Institute 2-shot adenovirus vaccine. And the symptomatic infection was reduced by 91.6%. And again, complete protection from death and severe disease. So this is all fantastic news. We still don't know some things about the vaccines. For example, how well do they block transmission? How long the immunity lasts? What are the correlates of protection and protection against the variants? This is a major issue as, people in Brazil are facing the P1 variant that may have a reduced efficacy from some of the vaccines. And so this is something to really monitor and be proactive about protection. As I mentioned, the mRNA vaccine, actually all of these platforms are quite amenable to the production of the variant spike antigen vaccine. So I know that most companies are already making the variant version of the vaccine. So here's my ending message to all the young Brazilian scientists. This is a quote that I made, I guess. My student, Yeshin Yang, had made this beautiful drawing of me looking into a microscope and saying, to be successful in anything, you have to enjoy it and enjoy yourself while doing it. And I truly believe that. For me, if I didn't enjoy science, I wouldn't be able to do it well, and I wouldn't enjoy myself. And so really keep an open mind. Finding something that you love is an important part of this path. So learning basic sciences is incredibly important. Even if you want to become a biologist, you really need to understand math, physics, chemistry, and other disciplines in order to succeed in today's world. As I mentioned, we employ all of these techniques to improve our understanding of the immune system. Scientific training is all about how to think and not what to think. Obviously, what to think is what you want to do, but learning how to do science is really about how to think, how to formulate a hypothesis, and how to design an experiment to be able to get to the truth. And that's really more than half the fun of science. And obviously, finding an area you are interested in and dive in deep. This is obviously not a easy thing to do, but once you find that something that you love to do, that's you will know it, and you can dive in deep. So I want to end by thanking my amazing laboratory, who every one of them contributed so greatly during the pandemic, not only in the immunological analyses, but also in helping each other. They were doing night and day PCR analysis for helping to detect COVID cases in the community. Also want to thank the amazing team of investigators that allowed us to collect these samples called Impact Yale, which is headed by Dr. Albert Koh, as well as many others who we collaborated with. The study that I described earlier was headed by Carolina Lucas and many other authors who contributed, and the funding agencies that allowed us to do this research. So I'm going to stop sharing now to take questions.
Thank you so much, Akiko. It was really an impressive talk, really broad, and I guess exactly what we needed right now. As you know, Brazil is exactly in this moment in a very critical situation. So I guess understanding more always helps. We have tons of questions, so I have to say now that I will not be able to ask all of the questions. I apologize for that in advance because Akiko's time is limited, but we will ask a few questions. So the first one we have is from Rodrigo Figueiredo. He's asking, do you think that there's a role for co-infection in severe COVID-19 as for influenza bacterial co-infection? Invasive aspergillosis has been also observed in patients with severe COVID-19.
Yeah, a really good question. So, before COVID, we were studying influenza viruses, and essentially, most of the severe and lethal influenza virus is caused by a secondary infection with a bacteria. And so we were expecting to see a similar, proportion of people having co-infection with COVID, and we haven't found many. There are some instances of co-infection that leads to severe, disease outcome, but it really isn't required to elicit the severe diseases that we're seeing. So it's fundamentally different from the influenza case.
Okay. We have another questions about vaccines, of course, and as you may guess, we're very preoccupied here with the variants.
Mm-hmm.
So Gabriel Luis Moreira is asking, which kind of vaccine would be better facing new variants? And I guess he's referring to the fact that, as you know, in Brazil we have the spike protein specific vaccines, but we also have inactivated vaccines, the Chinese vaccines. So one of these seems better for you at first, or not necessarily?
Yeah, I haven't seen sort of variant-specific protection data from the inactivated vaccines yet, so it's hard for me to recommend one or the other. It's really important. What I try to emphasize was that all of these vaccines will hopefully prevent severe and lethal COVID from the variants as well. So far, everything that's been looked at suggests that. And so the important thing is to get the vaccine as soon as you can. Obviously, it's not widely available in Brazil yet, but once you have the opportunity, I would take anything at this point, like anything that's been approved for use are quite effective in preventing severe or lethal COVID.
Yeah, that's an important message. Don't choose your vaccine. Just go and take whatever is there because it will protect you to some level. Mm-hmm. A question from Adriana Bonomo. How do you put together the findings of anti-IFNI antibody and disease severity given your data showing that high IFN response associated with disease severity?
Yeah, that's a good question. I took out a mechanism slide, unfortunately, but essentially interferons are incredibly important during early stage of infection, as I mentioned. If you don't have this innate resistance mechanism, you will have a very high titer of virus and suffer from severe COVID. And that's what's happening with the anti-interferon antibody that's seen in severe patients. However, some people basically have very low interferon, they let the virus replicate, and then starts inducing interferon later during the infection. And that also has a harmful effect. In fact, animal models that were tested for the MERS virus by Stanley Perlman's group showed that injecting interferon 2 or 4 days after the infection killed the animal, whereas injecting interferon early or prior to infection prevented the death from animal. So really, it's the timing of the interferon that matters. And so late interference is probably harmful. Early interferon is absolutely needed for clearance of the virus.
Okay. Another question from Nathan Berkowitz. Could you explain if useful data comes from the first testing vaccines in other species?
Useful data?
Yeah.
Oh, well, yeah. So every
to test these things in other species, or could we have gone directly to? I mean, explain better how this whole thing works. You know, testing in animals first, and then.
Yeah, absolutely. So we always do stepwise analysis of a vaccine, usually starting from a small animal model like the mouse or hamster or something else, stepping it up to the rhesus macaque model and then human model. And so, as I mentioned, the mRNA vaccines have already been tested in multiple different diseases. And so they've already undergone all these kinds of testing before. But yeah, if you were to come up with a new vaccine, you always go from the small animal model, larger animal, to human clinical trial. But this time, with the COVID-19, things were done very quickly and in parallel sometimes.
Right. All right. I'm running because there's so many questions.
I'm sorry.
We'll leave you. We'll just take a few more. Okay. Daniel Bargari is asking, could you comment whether inactivated vaccine could be working through neutralizing antibody even with low titers, or other mechanisms are involved, T cells, non-neutralizing antibodies? I think it is important in light of the VOC, I don't know what that is, and loss of neutralizing antibody activity seen in VNT for these vaccines. It's those are immunology jobs.
Yeah, sorry. That's the variant of concern on the VOC. Okay. So yes, inactivated viruses elicit not only neutralizing antibody, but also T cell immunity to some extent. As I mentioned, both antibodies and T cells are important for preventing spread and replication of the virus. And so the one advantage of the inactivated virus is that it contains not just the spike, but all the structural component of the virus, right? So all the other proteins like the nucleocapsid, the envelope, the matrix, it's all in there. So you might also generate antibodies and T cells against these other components that may help counter the variant, because the variants do have mutations in some other genes, but are not going to interfere with obviously all the epitopes that are in conserved regions of the nucleocapsid, for example. So, there is pros and cons to every vaccine type, but I'm hopeful that the inactivated vaccine can induce a more broader immune response as well.
Right. Okay, I'm going to take two more, and then I'm sorry, I have to stop because otherwise we'll spend the day, and that couldn't be possible. So Luisa Ramirez is asking, in the first part of your talk, you mentioned the possible identification of specific genes helping the COVID virus spread in the body. How this identification would help developing new treatments or methods to attack the virus?
Yeah, good point. So in fact, I didn't present the data itself. I just summarized it. But what we found was that when you take, we took 71 different kind of proteins in the plasma of patients, and we asked like an unsupervised clustering of patients based on that 71 factors. And it created 3 different kinds of clusters. And the first cluster of patients had enrichment in tissue repair factors, such as growth factors. And those are the patients that ended up recovering from this infection. And they had, essentially a moderate and then mild disease. Whereas patients who had elevated levels of these mixed cytokines were the ones that developed worse disease. So this led us to the understanding that, this sort of mixed cytokine response is maladaptive. And if we can focus our immune response to generating these tissue repair factors, perhaps we can help people recover faster. And so even though we don't have a therapy yet, the insight might help come up with a better therapy to increase the sort of tissue repair function that allows the people to recover faster.
Yeah, that's true. All right, Danielle is asking, a recent paper showed that asymptomatic patients' interferon and TNF response are high, but in contrast to what happens in symptomatic patients, this is accompanied by high IL-10. Could you comment? That's very specific.
Yeah, so if I can speculate what might be happening. Yeah, so like I mentioned, the timing of cytokines is key, right? So the same cytokine can either save or kill you depending on when it comes up. So in the asymptomatic patients, as I mentioned, if you are to generate robust interferon early, you will, basically be able to clear the virus and have very mild to no symptom. Whereas, if you elevate it later, it might be toxic. But in addition to the interferon, the IL-10, which is an immunoregulatory protein, may, either dampen the inflammation or contribute to further replication of the virus because it's dampening the immune response. So again, the timing of all of this is absolutely key. And so we really have to think about the cytokine as kind of an orchestrating a response at different time points. And so we would really need to look at the timing of all this.
Right. Okay, last questions, guys, because then we'll have to let Akiko go. A question from Andres Guzman. Do you find it challenging to present the importance of basic research to the general public? That's a more broad question.
Yeah. I used to, I was a little bit, well, I was, before the COVID, I wasn't this public in terms of my science communication activity, but I felt that it's a duty of a scientist to communicate the truth. There's so much misinformation out there, and people who are, don't have the background really do not know how to evaluate these things. So I decided to be brave and just start communicating science to the public. And the more I do it, the more I get used to it. And I think I'm getting better. But it's really about practicing, yeah.
Yeah. Well, Akiko, one thing I have to say is that communicating science is something that we do, and so we cherish a lot at Serrapilheira. I think it's great to have people like you who can tell you what we know and what we don't know in a humble way. Because what we've seen many times is people speculating, and that very often in this type of crisis doesn't help much. So being humble and give the data, this is what we know, this is what we still do not know. And I really appreciate the fact that you do this in a very clear way. The other thing that I found very important is you emphasizing the role of women in science and showing very clearly that sometimes they've been put aside, and that's just not acceptable anymore. So I guess more people need to talk about this, because the 2 examples you gave are not the only one, and they're very various. The role of basic science, obviously this RNA vaccines didn't come out from nowhere. As you say, you cannot just inject RNA in an arm and it will give, those are lots of work. Perseverance, I think, is an important aspect. You have to believe in your idea. You have to persevere, and otherwise things just don't go through. And so sometimes you get your grants rejected. Sometimes people don't believe in what you do. And there's this one aspect of believing in what you do. I think that's very important. Finally, I want to thank you because you are in our board, and having spent 1 hour of your time with us today, which is a very special, a sad day in a way in Brazil, but I guess the best response is science. We know that. So having you today was a privilege. I want to thank you again, Akiko, for your presentation. And again, I want to remind all of you that you can apply to the program, and the application is on March 9th. So please be ready, because we will favor the first 500 inscriptions on March 9th on the website of Serrapilheira ICTP. You can find all of the informations, and you know, this is going to be a great course. All right, Akiko, thank you so much for your time.
Thank you so much, Hugo. And I'm really looking forward to the launch of the training program.
Thank you. Bye-bye.
Bye-bye.
Bye-bye.